Okay. So one of the major uses of bioinformatics APIs is to use them to integrate analysis from external sources into bioinformatic workflows. So for example, we can perform term enrichment for differentially expressed genes in Enricher, tissue expression for a single gene from Gene Ranger, obtain enriched kinases from differentially phosphorylated proteins from KIA3, or enriched transcription factors from differentially expressed genes from KIA3. We can also find genes that are co-mentioned with a biomedical term or augment a gene set with co-expression from gene shot. And another thing we can do is to use sitcom links to find the mimicker and reverser signatures for our analysis signature. I've linked each of these web pages and tools that I talked about in this description tier. So let's go through like an example workflow. We can start a workflow from a study that compares a cohort with type 1 diabetes to non-diabetic controls to find that alpha cell function and gene expression are compromised in type 1 diabetes. GEO, or the Gene Expression Omnibus, is a major open biomedical research repository for transcriptomics and other omics datasets that currently contains millions of gene expression samples from many studies collected by research labs across the world. And so we can obtain the samples gene expression samples from D2H2, which serves transcriptomics datasets created from GEO that are relevant specifically to diabetes. We can then perform differential expression analysis in Python using DSEQ2 in order to find differentially expressed genes from between these different experimental conditions. After extracting the significantly up and down regulated genes, we can then submit these genes to Enricher in our workflow using the Enricher API to find enriched terms or pathways for these up and down gene sets. So just looking at what the code would look like for the first part of the workflow that I described. First, you would be importing the S3FS, or which is a Python file system interface for S3, and then importing each of these packages, which will be important for loading our data set and also for performing the DGE analysis. So firstly here, our data is stored in an S3 bucket and for D2H2 given by this endpoint. And so we would use the S3FS package in order to load these files. So the way it's stored in the bucket is the space first of all, we have the space URL, then we have the species that we're trying, the species of the study, and then the GSC. And so to get the expression file, this is the expression.tsv file, and the metadata file is the metadata.tsv file. And then so after opening up this particular file given by this path, we then load this into a pandas data frame using the pd.read CSV. So now we have accounts data frame, which is containing under all counts for the study, and the metadata data frame, which is containing the sample metadata. And so to perform the differential gene expression analysis, we would first have to load each of these files into the DSEQ dataset class. And so the parameters for this is counts, but the way this class wants the counts to be structured is each gene has a column, and then the rows are the sample barcodes. But the way our counts account's data frame is structured as read from the S3 bucket is in the opposite way, with the genes as the indexes and the columns as the sample barcodes. And so we transpose this file first. We then put in the metadata data frame into the metadata field, and then the design factors is the name of the column of the metadata to be used as the design variable. And for us, this is the condition column. After creating this class, we would then call this DSIC to a wrapper, which is a wrapper for the first part of the DSIC2 analysis pipeline. And this helps to perform the dispersion and the log fold change estimation. So after this, we would then be calling the DSIC stats, which would implement the p-value estimation for the differential gene expression according to the DSIC2 pipeline. From here, we would then be able to get our p-values, adjusted p-values, and our log to fold change. We can then view a summary of what this data frame would look like with each gene and its p-value and log to fold change and other statistical analysis scores using the dot summary function. After this, we can then basically create a data frame that would sort the values based on the adjusted p-value, which is the p-adj column. After we perform this analysis, in order to get significantly differentially expressed genes, we would use a p-adjusted p-value criteria of it being less than 0.01. And so this is essentially just extracting all the genes in our data frame that have an adjusted p-value of less than 0.01. The up genes would then be the genes in this filtered data frame that have a log to fold change greater than 0. And the down genes would be the genes that have a log to fold change of less than 0. We can then see that after we've gotten our up and down genes, we can then define a function to submit these differentially expressed genes to enricher. And so here's like very similar to how we wrote our own function earlier. We're just creating a function that will take in a gene list and its description, and then make a post request to enricher using the add list endpoint, so that we're able to get a unique ID to append to the enricher, to append as the dataset query parameter input to this link, so that we're able to view our enrichment analysis results in the browser. And so after making this post request and then retrieving the unique IDs for each of the up and down gene set, and then we then append this to the URL, which is enriched, and then with the dataset query parameter, we then see that clicking on each of these links will then take us to the enricher page, where we're able to find to view the enriched terms from each of these different gene set libraries for each of those gene sets. And so in order to get the enrichment and analysis results programmatically instead of just viewing them in the browser, we can also modify our enrichment function to also call the enrich endpoint that we talked about earlier to extract the enriched terms for a specific library. And so here we see that we add a new input parameter to make for the function. So here, instead of just appending this user list ID that we get into the enrich to view it in the browser, we then call the request.get and the enrich endpoint using this user list ID. So and then specifying also the background type as the library that we want, so that we are able to retrieve the enriched terms for that specific library using this get request. And so here we see an example of what that looks like when using the down gene set and using the Reactome 2022 library. The format is pretty similar. Oh, yes, Hisu?
Yeah, so we are getting close to 2:30 and like there's some more materials we need to cover.
Mhm.
So once it hit 2:30, feel free to leave and Dr. Mayan will post the recorded video rest of the session. I just shared the link on the chat, so yeah. And Stephanie, please go ahead and yeah.
Okay. And so here we see that using this particular function, so and making another additional GET request after receiving our user list ID with the enrich endpoint, we're able to view the enriched terms for the Reactome 2022 library in our code without having to view that in the browser. And so although now we have our enrichment analysis results from Reactome, it's in a JSON format, and so this is just basically the key value mapping that was described earlier. That is not very clearly undefined, and so we can format our results a little better to make them more readable. And so to do this, we can create a pandas data frame with each column for a different part of the response. So a different column for the rank, a different column for the term name, a different column for the p-value, odds ratio, combined score, and so forth, and so forth. And here, the way we would do this, because we know the structure of our output is a dictionary, which is mapping the library to the list of lists results, we know that we're only getting one library. And so we would only have one key value pair. And so our data to our data frame would then just be the values, which would be the list of lists. But because we're only having one key value pair, we would just index to the first one, which is on the value for the Reactome 2022 library. And then specify the columns that we would want, which would be all these parts of the response. And then in order to make this a little bit more readable, we can then set the index of this data frame to be the term name. And this line of code is essentially just dropping the old p-value and the adjusted p-value. So these are not part of the final data frame that we're going to see. And so now we see that the data frame that is created, the enriched DF data frame, now has organized our results a little better. And so now it's more readable. So we have for each of the term names from the Reactome 2022 library, we have on the rank of the enriched term, its p-value, its odds ratio, its combined score in a readable table, which can be exported for other uses. We can then expand the workflow by using the SIGCOMM links API to identify the mimickers and reversers for the signature that we have obtained from our analysis. SIGCOMM links is a bioinformatics web app that serves over 1 million gene expressions signatures processed, analyzed, and visualized from links, GTEx, and GEO. And so given a set of up and down regulated genes, SIGCOMM links can perform a signature similarity search to find mimicker and reverser signatures. A mimicker is essentially a signature that would have similar up regulated genes to the input up gene set, and then similar down regulated genes to the input down gene set. A reverser is the opposite, which is a signature that essentially would have similar upregulated genes to the input down genes, and then similar downregulated genes to the input up genes. And so now we can utilize the SIGCOMM links our up and down genes for a signature search. So the API is linked over here. And firstly, we would need to specify the metadata API, which is this. And then for every gene symbol, the way the SIGCOMM links enrichment analysis works is that we would need to find the gene ID of a specific gene in our gene set, and that is what we will be sending in for enrichment. And so first of all, let's make an input gene set object, which is a dictionary that contains upgenes and that will contain our upgene set in upgenes and downgene set in downgenes. We can then just merge these two gene sets together so that we can pass it into a post request that is asking the SIGCOMM-Links API with this entity/find endpoint to get the gene ID of every single gene that is in both our that is in our up and down gene sets. That is what this payload is describing. So using the filter where we're finding return every gene object in the SIGCOMM-Links database that has the same meta dot symbol as the gene in our gene set. And so after doing this, we get our response, which would be the gene objects as defined in SIGCOMM-Links, which would be the ID and then the ID and then the meta dot symbol of that gene object. And then so that we're able to get back our signature defined by our up gene set and our down gene set. So now we want to get two different lists. One list being the up entities, which would be all the gene objects, the SIGCOMM-Links ID for that gene, for each gene in the up gene set, and then that for the down gene set. To do this, all the entities that we got from our initial post request here, we will then be looping through each entity and then checking. So if the gene symbol of that entity is in our original up gene set, add it to the up entities here. And then if the symbol was in our original down gene set here, add it to the down entities. So now we have our signatures, but instead of being defined by gene symbols like they were defined here, they're now defined by the gene entities that as they're saved in SIGCOMM-Links. And so after this, we can make another post request to get the persistent ID to make a post request to basically add this user defined input to the SIGCOMM-Links database so that we're able to so that we get a persistent ID for this signature. And so here, that is what this post request is. We're doing request.post with the metadata API, specifying that we want to use the user input endpoint. And then sending in this payload, which is saying that we want to add a new signature, and it's containing these gene entities. After which we can then get our persistent ID from this post request using the rest.json and indexing into the ID key here. This persistent ID can then be used to view our mimickers and reversers in the SIGCOMM-Links page by appending it into this URL, which is the signature search and /rank. An example of what this would look like would be after running this function, we input our up and down genes. We're able to get this URL, which would display the mimickers and reversers for our differentially expressed up and down genes in this web page here, as shown. And another thing that we can integrate into our workflow is submitting the gene sets to GeneShot for augmentation with co-expressed genes from ArchS4. GeneShot is another one of the tools that the lab has created, and it's an application which converts PubMed identifiers or PMIDs returned for arbitrary search terms into a ranked list of genes using gene publication associations, such as those encoded in GeneRiff, which is the gene references into function. So GeneRiff is essentially a file which maps PubMed IDs into maps genes into the publications that they are found in. And so gene set augmentation is the process of adding genes are likely relevant to an input gene set based on gene similarity matrices. And these matrices can be defined by gene co-occurrence matrices, and which basically are a matrix that would define um, how much specific gene is occurs with another gene. And this we can also use gene co-expression data from the ArchES4 resource to construct these gene similarity matrices. Another way to perform gene set augmentation is to use the protein interaction networks, and so genes that are in the same network as a given gene set. The addition of related genes can be used to capture subtle but biologically relevant changes in expression patterns that might be missed by using traditional GSCA. And so using the gene shot documentation, we can structure our API call by replacing the documentation gene set with our gene set variable, and then selecting co-expression for the similarity field as we want to augment our gene set with co-expressed genes. After calling the co-expression augment function here, which essentially is performing the post request in order to retrieve these co-expressed genes based on our gene set and based on this similarity metric, we can then extract the co-expressed genes by looping through each of the values found in this association key value pair and adding it to our final gene set using list comprehension. And so here we know that the structure of our output from the post request is structured like this. And so we index first into association, and then for each item in association, each gene, we are then adding it to our final gene set. And so we do the same thing for the down genes and then the up genes. And so now we finally have now we finally have an augmented down gene set and an augmented up gene set. And so we see that here with augmentation with co-expression, we're able to make our up gene set go from 341 genes to 541, and our down gene set go from 132 to 332. We can also submit the augmented up and down gene set to enricher for enrichment analysis using the submit to enricher function that we defined earlier. And so now we pass in the up augmented genes, and then we also change on the name so that we're able to differentiate between this and our original up gene set, and do the same thing for the down gene set. And so this function would return to us the unique ID of that gene set after it's been submitted to enricher and added to its user gene set table in its database. And so after retrieving this persistent ID for our up and down gene sets, we can then append them to this link to the enricher to this link where with the data set query parameter so that we can view these gene sets in our browser, as we see at the bottom. So here we have the up augmented gene sets, and we can see the enriched terms for each library, and we have the down augmented gene sets, so we can see the enriched terms for the different libraries on the enricher page. If we would like to get these enriched terms programmatically, we can do the same thing that we did for original gene sets similarly, that after we've gotten this persistent ID, we then do the request dot get using the enrich endpoint and specifying the background type that we would like, and then using that to find the enriched terms for that specific library. Finally, to streamline our analysis pipeline, we can parameterize our workflow such that for a given study, we perform all the steps in using a single function. And so firstly, we can combine our data and metadata extraction for the study and the the differential expression analysis into one function called D2H2 DEG. So this would take in the GSC study and the species, and then we would then be able to extract this from D2H2, like we described earlier, as the first part of our workflow, perform the differential expression analysis, and then get our up and down genes. But specify putting this into one function now. And so all we have to do is specify the GSE and the species, and then we're able to run the single function in order to get our up and down genes. We can then call this D2H2 DEG function here in our main function. And after getting our up and down genes, we can continue our workflow by augmenting this if the function input is true. If we then go in and augment these genes and then submit that to enricher. And then we could also then submit this to syncom links using the syncom up and down genes function that we talked about earlier, which would take in the up and down genes and then use the syncom links API to get the mimickers and the reversers. After which we can then call again the submit to enricher library function, which instead of viewing this in the browser, like we have for the submit to enricher function, we would be able to see our result programmatically for a specific enricher library that's specified in the input to this function as well. And so that is kind of how we would join all of our steps into one single function called the analyze D2H2 study. So here, if we call this function, specifying a specific GSC and a species, and we can then specify whether what library we would want to use as our background, and whether or not we would want to do gene set augmentation before our analysis pipelines. This single function would then be able to perform our entire workflow one after the other and performing multiple API calls.